Hello everyone, my name is Murtura Enoch and we are going to revise together BI 110, and the question reads 1A. Discuss the role played by the following parts of the cell. Roman numero 1, centrioles. Roman numero 2, smooth endoplasmic reticulum. So one thing that you should bear in mind is that whenever you are answering, what should guide you is the marks that are being given. So we can start answering. So we have 4 marks, so that simply means that on each part of the cell you should bring 2 points so that you obtain 4 marks. So the first one, that is centrioles. So one thing that you should bear in mind is that the role of centrioles is that they are responsible in formation of spindle fibers. So they are responsible in formation of spindle fibers that facilitates movement of chromosomes. So they are responsible in formation of what? Spindle fibers that facilitate movement of chromosome during what? Cell division. The second one is that they are responsible in formation of cilia and flagella. So you writing these 2 points, it means that they are going to award you 2 marks. So we go to the second one, that is smooth endoplasmic reticulum. So one thing that you should bear in mind is that in smooth endoplasmic reticulum, that is where there is synthesis of lipids. So synthesis of lipids and steroids. Another one is that it also helps in regulation of what? Calcium ions that are responsible in building up of bones. So there are various points that you can bring, or the role of the same parts that have been mentioned. So you can also say that they are responsible in intoxication of what? Poisons. So these are the required solution. We proceed to the next one. So the second one, they are saying distinguish between prokaryotic and eukaryotic flagella. 4 marks. So there are various ways of differentiating, but it's very cardinal to just write them in the table, or in this way that I'm going to represent it. So what you should know is that prokaryotic flagella, they use proton motive force, and then eukaryotic flagella, they use ATP. So prokaryotic flagella, they're supposed to be use protein motive force. Another one is that when they are moving, so they rotate in movement. This one they do what? They bend in movement. Another one is that prokaryotic flagella, they contain flagellin protein. Wow, these eukaryotic flagella, they contain tubulin. They contain tubulin protein. Another thing is that their base body are anchored. They are anchored in the cell plasma or envelope. These ones, their body plasma, their base body are anchored in cytoplasm. So you're writing like this one, it means that you're going to obtain 4 marks. So from there we can proceed to the next one. So see, they are saying describe how the differential interference contrast microscope work. 4 marks. So one thing that you should know is that the differential microscope operation, the other name is Nomansky optic. So the same microscope, one thing that you should bear in mind is that it uses polarized light, that is just a light wave in which vibration occurs in a single wave or in a single plane. So polarized light, that is just the light wave in whose vibration occurs in a single wave. So it uses polarized light. So when light passes through the specimen, it splits into two beams and recombines when passes through Nomansky prism, creating a contrast based on their diffractive index, and the 3D image is formed. So that is just the operation of what? Normatic optical or differential interference microscope. So we proceed. They are saying give 3 reasons why mitochondria and chloroplasts are considered to have existed on their own as independent. So one thing that you should know is that mitochondria, they are considered to be on their own. So mitochondria and chloroplasts, the reason why they are considered to be independent, because mitochondria and chloroplasts have their own DNA that is circular. Another thing is that mitochondria and chloroplasts have, or they divide, so they divide by binary fission. Another one is that they have their own ribosomes that are what? 30S and 50S. So please don't forget to subscribe and like the video, also share. So see you in part 2 or question 2 of revision of BI 110.